Okay, now I'm welcoming the team from Munich. Please give them a round of applause.
Hello, everyone. We are Team Munich, and we are representing InkSight. InkSight is an optical biosensor in the form of a tattoo. It's a biological ink composed of engineered mammalian cells containing synthetic melanosomes. The cells are embedded in a hydrogel in the dermis. The cell allowing for detection of biomarkers in the interstitial fluid. Upon biomarker detection, a cascade of reactions will lead to a contrast change and an expression of our pigment, making the tattoo visible for our user. We focus on a broad range of targets, from hormone tracking for cancer monitoring and therapeutics to heart disease and beyond. The advantages of our system are continuous monitoring, minimal invasiveness, modularity, and a visible readout for patients. We have 3 distinct components to our system. The first one is our receptor, MESA. The second one is the enzyme tyrosinase, and the third one, encapsulants.
Thank you, Kalina. Let's move on to explaining MESA. So the way MESA works is we have a ligand with 2 epitopes. It can also be a dimeric ligand. We have 2 chains, each has a ligand binding domain, and they bind the target separately. This induces dimerization of receptor, and a TEV protease can reconstitute. This TEV protease can then lead to downstream signaling. For our software, we designed a software called MESA Designer. With this software, you can choose any targets, design the receptor in minutes, and arrive at a finished construct. Advantages of the software are that you have blazingly fast deployment. It's experimentally validated. You can export it directly to a GenBank file and then use it in downstream processing. We offer a Python API. We offer a Python package. We offer a web application which you can use on desktop and on mobile. For the validation of the software, we conducted wet lab experiments. Here we have a MESA receptor, which then reconstitutes the TEV. The TEV activates a TTA transcription system and expresses a reporter YFP protein. For the software, for the validation, we can see here that we successfully were able to induce expression of a reporter upon adding the IgG1 FC domain, which was the target we designed it for, and we observed that expression ratios increased by 160% for the MESA receptor. So you may ask yourself, how can we actually change the color? We do this via harnessing one enzyme. It's called tyrosinase. Tyrosinases catalyze reaction of L-tyrosine to L-dopa and then to L-dopaquinone. L-dopaquinone then can spontaneously react to eumelanin. Eumelanin, or melanin, is a very potent and dark pigment with a very broad absorption spectrum, making it suitable for visual output. For activation of this tyrosinase, since we want to make the system activatable, we engineered a tyrosinase that has a natural inhibitory domain and exchanged the protease site for a TEV cleavage site. Here, we can see that adding the TEV protease that cleaves off this inhibitory domain increases the production of melanin 6-fold. Because the intermediates, such as L-dopaquinone, are cytotoxic, we have to make this reaction take place in an encapsulated environment that is shielded off from the cytoplasm. For this, we let the tyrosinases integrate into nanocages, which then produce the melanin. One experiment, we could show that we can induce the forming of encapsulated nanocages, where we have the small monomers, where when inhibitory domain that inhibits the that inhibits the assembly of this nanocage. Here in the native page, you can see that without TEV proteases, we only have monomers, and with added TEV proteases, it actually assembles, and we only have fully assembled nanocages. Furthermore, something completely novel which we have done is we have fused tyrosinases to these encapsulants. And there we could also show that these assemble, which is pretty astonishing since tyrosinase are approximately 3 times the size of single encapsulin monomers. Additionally, we could show that our fusion encapsulins are 5 times as efficient in producing melanin as our positive control, which uses a different approach.
Now that you have heard about the 3 distinct components of our system, we can dive into the 2 distinct mechanisms for our contrast change. The first one is the simplest and slowest one. Activation of our receptor triggers the expression of encapsulin tyrosinase fusions that will assemble to functional nanocages and host the pigment synthesis. This slow mechanism is particularly suited for detection of biomarkers with slowly changing markers such as progesterone. For some markers, however, like for example, EPO therapies, you need a quicker response time. This is why we developed a second mechanism for contrast change, where all relevant components are continuously expressed in an inactive form. Here, the activation of the receptor leads to cleavage of all inhibiting domains and also allows the assembly of the synthesis to start. Our multidimensional project led us to engage with various stakeholders well beyond academia, from patients who inspired our troponin research to the public whose survey responses highlighted progesterone as one of our most important and popular biomarkers, and to professionals in safety, law regulations, and ethics that helped us develop our policy analysis. We also collaborated with a lot of inspired artists, creatives, and tattoo artists that inspired our very own art competition. Here, artists had the opportunity to basically submit designs on how they visualize our system to look like in the future. And we are proud to announce the winner, which was an anonymous artist with their amazing design of a shrimp tattoo. Here you can see how basically our system could be connected to a normal tattoo. The shrimp itself is a traditional tattoo with traditional ink, but the whiskers of the shrimp are our biological ink, which are going to appear once the biomarker is in very high concentrations in the interstitial fluid. Thank you, and this is InkSight.